Hello friends, in this session we are going to see some questions on embryology which are important for our university exam. So let's get started. Define embryology. Study of development events beginning with fertilization of an ovum and culminating with birth of a baby. Generally divided into two phases. Embryonic period from fertilization to end of 8th week. Fetal period from beginning of 9th week till birth of baby. Okay. Define fertilization steps and results. Fertilization is a process of union of male and female gamete. Steps in brief are penetration of corona radiata, penetration of zona pellucida, fusion of cell membranes of sperm and oocyte, completion of second meiotic division of secondary oocytes, formation of male and female pronucleus, fusion of male and female pronucleus. Results: just after fertilization, formation of zygote, restoration of 2n chromosome or diploid number of chromosomes, determination of genetic sex, initiation of cleavage, variation of species. So here you can see vitelline layer of protein receptors, sperm's head, mitochondrial material, nucleus, actin, acrosomal granule, jelly coat, fused plasma, egg plasma membrane, perivitelline space, cortical granule content, egg cytoplasm. Short note on spermatogenesis. Spermatogenesis is a process of event by which spermatogonia are transformed into spermatozoa. Stages: spermatogonia, 2 primary spermatocytes, each primary spermatocyte, first meiotic division, then secondary spermatocytes, each secondary spermatocytes converted into spermatids by second meiotic division. Each spermatid is transformed into spermatozoa by process of spermiogenesis. So here you can see Golgi apparatus, nucleus, mitochondria, centrosome, spermatozoon, acrosomal cap, head, neck, spiral mitochondrial sheath, middle piece, annulus, principal piece. Short note on spermiogenesis, process by which spermatids are transformed into spermatozoa. Following changes takes place in spermatid. Nucleus become dense and more ovoid to form head. Golgi complex, acrosomal cap, centrioles divide into 2 centrioles. Cytoplasm set off 1 to neck to form axial filament, 1 or more distally to form annulus. So here you can see spermatogonium 2n, first meiotic division, primary spermatocyte is 2n or diploid. It undergoes first meiotic division to produce secondary spermatocytes which are haploid n. Second meiotic divisions to form spermatids which are having haploid content. And here you can see spermatozoa are formed by the process of spermiogenesis. Short note on oogenesis. It is a process by which oogonia are transformed into oocyte. Oogonium enlarges to form primary oocyte. Primary oocyte undergo first meiotic division to form secondary oocyte and a polar body. Secondary oocyte, second meiotic division, ovum and second polar body. Oogonium, primary oocyte, secondary oocyte and first polar body. Secondary oocyte undergo second meiotic division to form ovum and second polar body. Right. Short note on corpus luteum. After ovulation, the empty Graafian follicle undergo influences of LH, luteinizing hormone, shrinks and is transformed into a yellowish glandular structure called corpus luteum. Steps: granulosa cells and theca interna gets vascularized by surrounding vessels and becomes polyhedral. Later, a yellow pigment developed in these cells and these are called luteal cells. Fate of corpus luteum. If fertilization occurs, corpus luteum persists for 3 to 4 months under influence of human chorionic gonadotropin HCG called corpus luteum of pregnancy. Secretes progesterone for initial 3 to 4 months. And if fertilization does not occur, corpus luteum lasts only 10 to 14 days. It degenerates and transformed into corpus albicans, a white structure, white scar, called corpus albicans of menstruation. So here is the pre-ovulatory follicle rupture, increase in LH, FSH, estrogen, inhibin decreases, proteolytic enzymes increases, kisspeptin increases. So ovulation happens, and in this ovulation secondary oocyte is released, and this graphene follicle undergoes transformation to form corpus luteum. Luteinization changes on cell cycle protein. So here you can see short note on implantation. Embedding of blastocyst in endometrium occurs 6 to 10 days after fertilization. Stages: zona pellucida disappears, trophoblast adheres to endometrium, trophoblast differentiate into cyto and syncytiotropoblast. Syncytiotropoblast penetrate the endometrium, migration of blastocyst into endometrium. So here you can see tropoblast, blastocyst, embryoblast, uterine epithelium, syncytiotropoblast, decidua, uterine glands, syncytiotropoblast, cytotrophoblast and syncytiotropoblast. Uterine epithelium reformed. Site of implantation: endometrium of posterior wall of uterine cavity near the fundus. Endometrium of posterior wall of uterine cavity near the fundus. Applied: if implantation is deep, it may lead to postpartum bleeding. Abnormal sites of implantation. The abnormal sites of implantation are within the uterus, near internal os, outside the uterus, uterine tube, tubal implantation, ampulla, infundibulum, interstitial part, rare. In abdomen, abdominal implantation, pouch of Douglas, retrouterine pouch, mesentery. In ovarian implantation. Sites of ectopic pregnancy: cervical, which happens less than 1%, interstitial 2%, ischemic, ampullary 70%, and fimbrial 11%, ovarian 3%, abdominal 1%. Small intestine, interstitial part, fundus of uterus, internal os of uterus, vagina, ovarian ligament, ovary, external os, fimbriae, infundibulum, ampulla, and mesentery. Short note on decidua: the endometrium of uterus that undergoes decidual reaction after implantation is known as decidua. Endometrium after implantation is called decidua. Decidual reaction: it occurs under the influence of HCG. The following changes occur in endometrium. Cells of endometrial gland swell due to accumulation of glycogen and lipid in their cytoplasm. Decidual cells. Nuclei becomes rounded. Number of cytoplasmic organelles increases. Function: it provides suitable site for implantation, to provide nutrition for early embryo, to provide immunological privileged site for conceptions. Parts: decidua basalis part towards the embryonic pole, that is deep to embryo. Decidua capsularis parts towards the embryonic pole, uterine cavity, it surrounds the embryo. Decidua paritalis. Remaining part of the decidua is decidua paritalis, and surrounding the embryo is decidua capsularis, and the decidua basalis is the part towards the embryonic pole that is deep to the embryo. Fate. Decidua basalis undergo development to form decidual plate within which give rise to maternal component of placenta. Decidua capsularis disintegrate and fuses with decidua paritalis and obliterates the uterine cavity. Decidua basalis, blastocyst, myometrium of uterus, decidua capsularis, and decidua paritalis. At the time of delivery, placenta separates with decidua and expelled out. Myometrium, decidua paritalis, decidua basalis, vitelline or yolk sac, umbilical cord, chorionic villi, decidua paritalis, amniotic cavity, chorionic cavity, decidua capsularis. Endocervical mucosa. Short note on blastocysts. The morula, consisting of 16 blastomeres, is transformed into blastocyst shortly after it enters into uterine cavity 4 days after fertilization. The fluid-filled space appears inside morula called blastocele cavity. As the fluid level increases in the cavity, the blastomeres are separated into two parts: trophoblast outer cell layer, inner cell mass, embryoblast. Human blastocyst, 5 days. Zona pellucida, trophoblast, blastocele, embryoblast. Fates. Embryoblast, forms embryo proper, trophoblast, embryonic part of the placenta. Fluid in the blastocysts provide nutrition to blastomeres. Go and download med notes. Thank you.